So thank you all for staying here with me before lunch and the organizers for the opportunity to present our work. So thanks, Hande, for introducing splicing and alternative splicing. So in our lab, we are very interested in studying alternative splicing in physiological processes and in disease. And in particular, we are interested in understanding splicing in cancer. So as she mentioned before, so there are in the many different splicing changes that have been directly linked to cancer processes, and they actually cause some tumor phenotypes. So as Abby mentioned this morning, so it's important to make clear what is here in for you, and I will start by basically saying what's in for us with this software that I'm going to talk about. So we wanted to analyze RNA-seq data from the Cancer Genome Atlas, and in particular, more than 5,000 RNA-seq samples, and we wanted to find an efficient way to do that, to study splicing with limited resources. So the splicing wish list is something, we need a method to study splicing accurately in thousands of samples with limited time and computational resources. And TCGA provided some information, but it was partial. Among other things, they were providing the transcript quantifications for RSM. So what do you do when you want to study splicing from RNA sequencing? So from the sequencing reads, what you want to basically measure is whether between two or more conditions, there is a change in the relative abundances of transcripts. Now, so there are multiple methods to do that, and in fact, there are multiple ways this method model this change in splicing. They are very different between them, from exon junctions or events-based methods and graph methods, or you can directly work with transcripts. So each one has advantages and disadvantages. I won't go into the details, but one common thing among them all is that they rely on the mapping to the genome. So that, although it's quite fast nowadays, it takes quite a lot of time, and you need a huge computational resources to do that in a reasonable time. So what could we do instead? Could we still use the information provided by TCGA and study splicing? So remember that, bear in mind that at the end of the day, splicing is nothing but the changes in the relative abundances of transcript expression that as Handes mentioned before. So an event or a junction or anything else, just a representation, a handy way of representing that and compute with that. So if we rely on the annotation, and then we can make a one-to-one correspondence of every event to the transcripts that describe the event, either that include or exclude the event, and then we can keep track of that. And if we have available the transcript abundances for any number of samples, so we can simply say that the relative inclusion is basically a ratio between the abundances of the transcripts that include the event or those transcripts that include or exclude the event. It's basically in line with the original definition of what the PSI or inclusion level is. PSI is basically a very useful measure to study relative abundances like Hande was mentioning before. Okay, so that's what we implemented in SUPPA. SUPPA is basically a software tool that, given an annotation, you can generate events from the annotation. It keeps track of which transcripts are explaining the event, and then given the TPMs or the transcript abundances for that annotation, it will produce the PSIs. And now that will produce it for a description in terms of alternative splicing events, the classical alternative splicing events. Okay, so the question was, does this work? So to benchmark this, we used first simulated data. So the simulation was simply using Flux Simulator with a given annotation. So the simulation provides a quantification of the transcripts. From that quantification, generate reads that you can use for your analysis. And then we use the simulated quantification only in genes with two transcripts to define, to give the standard PSI to be basically the relative abundances of the two transcripts. So that would be the value that we want to calculate. And the generated reads after the different simulations for the biases of sequencing were used to estimate PSI. So, of course, using the same mapping method for the mapping-based methods and using different quantification of the transcript for SUPPA. So we needed to compare estimated and simulated PSIs. And then we generally use two measures. One is a correlation, classical correlation. In this case, the Spearman's R between the two PSI values. And the other one is a measure of distance. It's using the absolute difference between the PSIs and the, sorry, this should be simulated, between the simulated and estimated PSIs and the cumulative proportion of that, so we can have an idea of how far, even though correlation might be tell you one thing or the other, how far we are from the actual true value of the PSI. So this was using the ISMB estimated transcript values plus SUPPA. So the question is how different is or how better or worse is compared to other methods. So at the time we were working on the paper, we used these different methods, RMATS, MISO, and you can see that RSEM plus SUPPA produces a quite good estimate compared to the other methods. Of course, you may think, well, but that's because you're using transcripts, and simulation was based on transcripts. But here's SG count, which is also a junction-based method. It's using grids mapped to junctions, and it's actually quite similar. So, and an important point for us was that if we use selfish, one of the earlier versions at the time, plus SUPPA, we can also get a quite reasonable good estimate. So that points into the direction of solving the problem for us of the speed and the compute. Another benchmarking was based on the RT-PCR. So reverse transcriptase PCR is basically that you use primers to quantify the transcripts that are produced from a given position in the gene. And you can do this quantitatively so that you can actually quantify the relative abundances in one condition or between two conditions. So we use the results, RT-PCR measurements from the same paper as Matt's paper in 2012. And the good thing is that they did the RT-PCR independently of long before we actually ran the analysis on the RNA-seq data, so that it's completely independent. Using these data sets, again, comparing with the same methods, we see a quite good or reasonable correspondence between what we can calculate with SUPPA, either quantifying transcript with RSEM or selfish, and with other methods. Okay, so some time has passed since then. There are faster methods, better methods. So we tried recently again the same thing with Salmon and Callisto to quantify transcripts, the same data sets, and we'll still see a good agreement with the RT-PCR. Okay, so I want to highlight this because that basically solves our problem. If we can quantify very fast with selfish, Salmon, or Callisto, then we can analyze splicing high speed. So is that true? So this is the timing for the mappings. That's the figures from the paper. So using, in this case, selfish, much faster than mapping reads to the genome. And then for the time of the PSI calculation, of course, we do some simple arithmetic calculation, so it's much faster. So could we solve the problem that we started with analyzing the cancer data? So hope yes. So we managed to calculate the splicing, the PSI values for all the events in the genome across the 5,000 samples. We linked that to the expression levels and motifs for RNA binding proteins, so we could calculate some links between RNA binding proteins and splicing events and come up with some hypotheses for a mechanistic regulation of splicing in cancer. And then, with the help with collaborators, we did some experimental verifications of these correspondences. So all these PSI values for all the events are available in Figshare. Okay, so now the question was, how about differential splicing? For this analysis of the cancer data, so we had to do comparisons between tumor and normal samples, or samples mutated or non-mutated for a specific splicing factor. So at the time, we having enough number of samples per condition, we simply use a classical method, a man-Whitney test with multiple test correction. But notice that to make it work or to make it reasonable, the results, we had to impose an extra cutoff to the delta PSI. And in fact, many methods, most of the methods, they force you to impose a cutoff, either delta PSI cutoff or a fold change like DEXEC or CAFTIF. So we were wondering, so how can we work with differential splicing if we have less samples per condition? How can we take into account this fact that the statistical test may be powerful enough to give you a significance, but the delta PSI is still not biologically significant. So that's our second splicing wish list. So can withstand SUPPA to do differential splicing accurately across multiple conditions? Why not, since we start asking? And again, limited samples per condition. An important thing to keep in the same spirit is that without going back to the reads, so that we don't have to rely on looking at reads that will take a long time. So to illustrate this, and what we're doing at the moment, I'm using data from David Elliott's lab, 2 conditions, 3 samples per condition, and there's a control and double knockdown of TRA2A and TRA2-beta splicing factors. I'll speak about another data set as well. So the idea is simply revisiting what Hande mentioned before, that there is a dispersion of the inclusion level of the transcripts or the expression as a function of the actual expression. So for delta PSIs, we observe the same thing. So these are the delta PSIs between replicates. So they are supposed to be the same, but they are not the same. And then as a function of the average transcript abundance of those transcripts that are explaining those events. So don't freak out. So most of the cases are here in between, although the plot is not good enough to show this. But most of the cases are around 0. But there are large dispersion. So that means that we need to take that into account for the differential expression. So if you do the same thing between conditions, then you see that there is also a dispersion in the actual differences that you see between the 2 conditions. So SUPPA for differential splicing does a very simple empirical test, which basically takes, for a given observation, takes the background distribution around that expression level, and then basically compares the observation with the empirical distribution of the background. And with that, what we find is, again, this volcano plot where you see the delta PSI as a function of the significance, and we see that the test separately separates well delta PSIs, but the small ones and the large ones. So how does this compare with other methods? So here we see RMATS. It's another method that takes multiple replicates. CAFDIF, DEXSEC, they all need this cutoff in delta PSI or fold change to be able to find the significant cases. And we see that RMATS also call significant things that have a very small delta PSI, so you need this extra cutoff. So we did also some comparison with RT-PCR. So these are 83 from this paper, and then we also had 16 from this other paper. Again, 2 conditions, 3 replicates each condition. So SUPPA, in this case, running Salmon first for quantification, and then SUPPA. So it stays quite good in terms of correlation, and also in terms of the distance, average distance between the RT-PCR PSI and the estimated PSI from the method. Now we are adding also MAGIC, which came up recently, because also takes into account 2 conditions and multiple replicates per condition. Okay, so here what we could see as well is we had a number of non-responders. That means they tried to measure by RT-PCR these events and they were negative, so we can see how many or what fraction of false positive we find for each method. So SUPPA stays more or less around the same percentage of false positives using 39 negative controls for other methods, and then also a similar set of true positives by using this. So we use multiple test correction per gene, and that's what we use here for cutoff as well. And again, the timing for the differential splicing analysis is much faster than other methods. So that this coupled with the previous quantification, which is very fast, I mean that you can analyze things in samples in a few minutes. So in this case, the data from Best et al. Was like 36 for PSI seconds, for PSI quantification, 6 minutes for the differential splicing. Okay, so let me finish quickly with how about the differential splicing across multiple conditions. So Pimentel et al. Paper, they also had data for different stages of differentiation, 3 replicates from each stage. So we wanted to know whether we can also work with these data sets. So SUPPA can basically, it will take all the samples, it will do comparison pairwise in the same way I just explained, and then it will generate the set of significant changes and a vector of PSIs, mean PSIs in each condition. With this vector, what we do is clustering. We can do an operation of clustering. We use density-based clustering because of 2 main reasons. So you don't need to specify the number of clusters, so that will be generated for you. And you only need to specify 2 parameters. One is the distance, maximum distance between vectors. that's very much like the difference between PSIs, that you allow events to be in the same cluster, and the minimum number of events per cluster, which naturally translate into the minimum number of events in a regulatory module. So it's a density-based clustering lends itself very nicely to the description of splicing regulation across multiple conditions. And we use two measures so internal measures for cluster consistency that has nothing to do with the biological problem that you are dealing with. So one is the silhouette score. I'll skip over this quickly, and the root mean squared standard deviation. So that will tell you basically whether with the parameters that you're using, your clusters are reasonably well processed. So, so this is using this data from Pimentel et al. Then we generated these two clusters. That's an example of what you can do with SUPPA nowadays. Okay, so let me wrap up. And okay, so SUPPA is much faster than other methods to calculate PSI values and differential splicing across conditions. And this is quite convenient for re-annotation and data reanalysis. So especially when you have updates, continuous updates with new data sets, that you don't want to spend weeks and, or months reanalyzing all the data. It shows good agreement with PCR validation, which is nice, so we can treat PCR as our gold standard. And it's very modular. You can actually apply the differential splicing or the clustering to any other of the outputs from other methods, like junction PSIs, isoform PSIs. You can use any other of the outputs, as long as you have the PSI values and a vector of values. Of course, the downside is the dependence with the annotation. So we don't see anything that is not in the annotation. However, there are some recent works in Arabidopsis, ARAPort11 and R2D2, which what they're doing is an effort to re-annotate the genome using multiple sources of data, improve the annotation, and then using Salmon SUPPA to quantify alternative splicing. I think that's the way things could go. So we improve the annotation and then quantify transcript with fast methods like Salmon, and then you can use SUPPA to do all the analysis. Okay, and of course, another downside of it is like you won't have time for a coffee break. Okay, so thank you very much.